All right, let's do it. I was gonna go over with you guys how to take notes because most of you guys are not doing it right. And that's probably why a lot of people aren't doing as well on the test as they'd like. If you find that you're writing down a lot, you're not doing it right. If you find that you are coming to lecture and listening to me lecture and reading the textbook, rewatching the lectures, doing this, that, you are not doing it right. You are doing too much, and you're probably doing it all like a night or two before the exam. You're not going to do well on the test if you do that. I was going to show you how to do notes appropriately. I left doing notes for this lecture, and I walked through with my other class, which is what I was going to show you, how I take notes for this particular lecture. And I had 3 pages of big with color diagram notes, and that was it. And it says less than what the study sheet says. Your notes should look like that. If you're writing more than what the study sheet says, you're probably either really not understanding anything that you're listening to that I'm saying, or you're going way too far, right? I'll show you what I mean. Because as we go through this, the study sheet, we'll start off with talking about what the difference is between sterilization, disinfection, antisepsis, and decontamination. If you were somebody who would be writing down every single word on this slide, wrong, 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 wasting your time and your paper and your ink and whatever it is that you're doing. All right. Sterilization, the destruction of all microbial life. You know what I would write on my notes? Sterilization kills everything. You know what that means, right? Why are you writing more? So disinfection destroys most microbial life. Well, that's not everything, right? Destroys less than everything. Okay, kills less than everything. Asepsis, also called degermination. It's disinfection on living surface. I would literally write disinfection on living tissue. And then decontamination, also called sanitization, the mechanical removal of most microbes from an animate or inanimate surface. So mechanical removal. Maybe you don't know what that means, and that's cool. So I would write a note extra about that. Mechanical removal is talking about, so I just put decontamination, mechanical removal, a little definition of what that means. Like scrubbing, and you're physically removing the microbes. You're not killing them and they're staying in place. You're physically removing them. Like when you wash your hands, when you wash your plates, scrubbing when you're prepping for surgery, filtering is separating microbes out from the liquid, right? They're being literally actually physically removed. So that is decontamination. That might be something you would write a little bit more about compared to other things. Okay, I have this muted and I forgot. Okay, so does that kind of make sense? Like you don't need to write down every word like this is the Bible. You write down what you need to understand the thing, right? Because when you guys go take the test, is the fact that you wrote down sterilization kills everything going to make a difference as opposed to sterilization, the destruction of all microbial life? Y'all in microbiology, you know what kind of life we're talking about. You don't need to be writing all that down. You already know. So that's just one example of like how you compare that down and how I would start off with, this is a lot of terms in this one. So breaking it down like that. Okay, let's go. This chart is excellent to come back to review which things sterilize and which things don't. Remember, sterilizing, killing everything. If you're not killing everything, you're disinfecting. If you are doing any of that on skin, asepsis, right? And if you're physically removing the thing, then that is decontamination. So you can see here, some of these things sterilize, some of them don't. In fact, what it comes down to is like when we are sterilizing something, killing everything in that whole area, typically you have to do it a certain way in order to actually achieve sterilization. So high concentrations of chemicals, because watered down versions aren't going to sterilize, but they'll be effective as disinfectant. Making sure you're exposing to heat for a certain amount of time to be sure you're killing everything off and all of that. So this is great to come back to when you guys are studying and you want to see what is overall going to fall into what category of disinfection and all of these other things, because that is really most of how, not everything, but most of how chapter 11's questions are going to go. I'll ask you about flutaraldehyde. Does it sterilize? That's what most of the questions are going to be like. Okay. All right, here we have our little chart of what is the most resistant. When we are talking about sterilizing, just okay, sure that whatever. Anyways, we're talking about sterilizing. We're more or less trying to include bacterial endospores, which you can see that at the top, they're the most resistant. Yes, I realize prions are there, right? But we'll get to that in a second. Bacterial endospores and everything below it. If you can kill endospores, you can kill everything below it. Okay, so if I tell you that a procedure kills endospores, it kills everything below it too. So that's where you can break down if you are sterilizing or not. Now, prions. First of all, these guys aren't really microbes, right? They're just misfolded proteins. So how do you kill that? You can't. That's not an option, right? They're not alive. They're just protein and misfolded. So these guys, first of all, they're extremely difficult to get rid of. Way more difficult than an endospore. But the problem with these guys also is that we don't see them like ever. They're super uncommon, very, very rare. So we don't typically think about them when we're designing, new ways of sterilizing things. So we're not thinking about that. We're thinking about endospores. Because where are endospores? If y'all think they're not everywhere, kidding yourselves. Even things like botulism. That's a Clostridium botulinum, which Clostridium and Bacillus, right? They make the endospores. So Clostridium botulinum is an endospore forming bacteria. There's a reason why people who brew beer sterilize their equipment. It's botulism because it's everywhere.
Isn't that also like?
Yes, because when I said the integrity of the can is broken usually, but yes, so there's a reason for that. And we can control things like that with things like pH as well. So we'll talk about this. We already know endospores are the hardiest. They are dormant. They are going to be able to survive a whole lot of really rough conditions. That's what they're built for, literally for that microbe to survive. So disinfection and antisepsis and typical decontamination, these are usually not going to get rid of endospores. So we have to go on to full-on methods that actually sterilize, kill everything. So again, sterilization, we're getting rid of everything. We're killing all microbes. Anything that we've done that to is called sterile. We would want to do this to our surgical instruments, syringes, and occasionally some packaged food. Disinfection, again, we're not removing endospores. That's what you should hear whenever I say that, okay? So it'll destroy vegetative pathogens, maybe not all even, but if it isn't killing all, but it's killing only some, disinfection, even if it only kills the gram-positives, disinfection, right? So a lot of times disinfection that's working appropriately will also remove toxins, which is helpful. Antisepsis, here we're going to talk about, we're talking about asepsis. This is anything that we're trying to keep microbes out of sterile tissues, like living tissues. So if I cut you open with a surgical knife, the tissues inside of your body should basically be sterile. So we want to keep it that way. So that is the process of asepsis. Sepsis is whenever you get microbes into those tissues where they shouldn't be. We know it's we say septic shock and having sepsis in general, that infection of being in the blood. Well, that's what that's referring to. So asepsis, we're trying to avoid that, right? Aseptic techniques, we already do this. We already do this in lab, right? This is the exact same aseptic techniques. We're trying to prevent contamination. And we might say, but we're not talking about living things, are we though? Because I guess in the way that you could think about it is the sample that you're working with is a living thing and you're trying not to contaminate it. So you could look at it that way, but anytime that we're trying to prevent contamination at all, that would be aseptic technique. We can particularly apply it to preventing vegetative pathogens on tissues. That would be antisepsis. This is not a weird word. I know it sounds like it is, but what are we talking about here? Antiseptics. That's not a new word, is it? So turns out you know that word. I would just write down antiseptics. You know what that is. Cleaning skin, yeah? That's what that is. Cidal. Germ, okay, you guys know what this is. Yeah, I don't need to really probably go over this. Cidal versus static. That's what this is going to be, these next two slides. Cidal kills, static inhibits. that answers that. So if you see bactericide, that is bactericidal, that means it is killing bacteria. That's all that means. If you're wanting to talk about general getting rid of microbes, you could say germicide or microbicide. Same thing with the static when you talk about that, inhibits growth, right? All right, then decontamination, we can also call this sanitization or sanitizers. These are often soaps or other detergent-based or work in a detergent-based way in order to remove the debris. All right, when you're trying to figure out how you want to control your microbes, you want to think about, do you need it sterile or is just vegetative pathogens okay? Are we going to use it once or discard it? Or like once and discard it, or are we going to reuse it? Will it withstand the procedures that we could be exposing it to? Is it suitable for a given application? I mean, are you guys going to roll your whole patient bed into an autoclave and autoclave it? No, right? That's ridiculous. So stuff like that. Will the agent penetrate to the necessary extent? Maybe you need it to go not just on the surface, but deeper in, and then the cost and all this. So this brings us to our medical devices and how we're going to group them based on what we need them, basically how sterile we need them. So if they're coming into contact with your sterile tissues, that's our critical medical devices. Our semi-critical, they contact your mucous membranes. It's like respiratory equipment, maybe thermometers. And then non-critical medical devices, this is going to be furniture, door handles, or like the machinery that's, running, the IV pump and all sorts of stuff like that. Okay, things that are going to affect how we're killing our microbes, the number of microbes that started off, the nature of the microbes in that population, type of microbial growth, temperature and pH, concentration of the agent, mode of action of the agent, and the presence of solvents. Basically, just talking about how this is going to work and how you have to think about it for each microbe. So if you're trying to, get rid of everything, then you have to think about how you're going to get rid of everything in the situation that you're going to be applying it in, right? And not just like one, we're worried about Clostridium. You want to think about more than that, probably. I actually cut this out of my other presentation, so I'm just going to pass it on. Okay, so there's different ways that these guys can affect the cell to help control everything, okay? It can target the cell wall, the cytoplasmic membrane, the cellular processes, synthesis processes. I don't like that. Just think of the nucleic acid synthesis, okay, of any of them. So we're talking replication and transcription for that one, right? And then proteins. So we're talking about the cell wall first. We're going to block its synthesis or we're going to break it down. That's the two ways we can approach it. Then that makes cells easier to lyse or break open. And then we have, this is the detergents and the alcohols that will disrupt the cell wall. Alcohols are pretty useful as far as they affect the cell wall, the cell membrane, and proteins if they're used correctly, but they still don't sterilize, surprisingly. So anyways, moving on. Then we'll have the cell membrane. Anything that disrupts the cell membrane, now we're having that loss of that selective permeability, the thing that kept things in and only let certain things in and out, right? Then vital molecules that are supposed to be kept in can go out, and we can allow entry of damaging chemicals. So a lot of times when we affect the cell membrane, we're going to include other chemicals that affect other parts of the cell too, to make it more effective. Surfactants are a group that falls within affecting the cell membrane. You'll see why in a moment. They're polar. They have a hydrophilic and a hydrophobic. You guys remember what that is? Yes, hydrophilic, like Philadelphia, love, right? Hydrophilic, brotherly love, right? Hydrophilic is water loving, hydrophobic, water fearing. We know a molecule that falls into this category, right? It's going to be the phospholipids that make up our membranes, yeah? So this is just like that as far as the structure goes. Not exactly chemically the same, but they have both of those regions. You guys remember the word for whenever they have both of those regions? Amphipathic, exactly. So good. So it's going to be amphipathic. Wow. So just literally mimicking our phospholipids. So now it's starting to make a little more sense. So look at the picture here. Our surfactants are in the teal. They can mimic, get in between, but because they're not exactly the same, they cause our disruption of our cell membrane. Make sense? So if I was taking notes, I would probably say disrupt cell membranes by mimicking phospholipids, because I know what all that means. You have to write down whatever makes sense to you. Anyways, microbial or nucleic acid synthesis and proteins. You need to obviously make copies of your DNA and go through transcription if you're going to be alive. So we can inhibit those things, and we can also inhibit translation by inhibiting the ribosomes. Remember, translation is making the proteins from the RNA. So we can inhibit the actual ribosomes from working and doing that. But there's another way that we can affect the proteins as well. So great, we know we can stop the DNA the normal way. With the proteins, remember we had talked about denaturing DNA with heat to separate it. So yeah, we should remember that because that's going to be on the lab exam, right? Anyways, the denaturing of proteins is the same thing. They're also governed by hydrogen bonds. So heat and pH can affect it pretty significantly. So that can denature or cause change in the structure of the protein, where that protein depends on its structure in order to do its job. So we're, messing that up. So how do we denature? Moist heat is pretty common. You guys ever cooked an egg? Then you can't go back from that, right? If I took your whole body and roasted it over a spit and let you cook down, there's no coming back from that, right? So it's pretty irreversible. And then we have our organic solvents. Alcohols can do this. Acids can do this. And then we have the phenolics. So phenol, which we'll talk about later, but yeah. And then other effects on proteins, metal ions can bind in the active sites. Right, so this is a picture of denaturation. The top, that like squiggly line looking guy, that snake looking guy, that is our denatured protein. We need to be folded in order to function properly. But we can also denature by just making it have a different shape. It doesn't have to completely denature. As long as it can't work on its substrate anymore, it's denatured. All right, let's talk about the physical methods of control. It's going to be heat, radiation, filtration. It says ultrasonic waves, but we're not going to talk about that. We are going to talk about osmotic pressure though, and then cold. Start with heat, moist heat first, okay? So heat can go two ways, moist and dry. Moist heat includes hot water in general, boiling water, and steam. Temperature will range from 60 to 135. I'm not going to test you over this. If it's not on a study sheet, guys, I'm not going to test you over it. So that's another thing too. As you go and make your notes, look at the study sheet. Did your note cover that topic on the study sheet? Like all that it's written there? Then check it off. You don't need to keep writing about it. That's another way you can look at it. Check it off as you go. So that's how I would do that. Note page and then study sheet and looking at this and making my notes and then checking it off as I go. If we come to a topic that I cover that you guys don't understand, that I may explain it, but you're still not getting it, you don't want to ask about it or whatever it is, you want to revisit it for whatever reason, circle it on your study sheet and then go look it up, ask ChatGPT, ask a friend and work it out, or whatever you need to do to understand that topic, then come add it to your notes, okay? Anyways, so yeah, we want to use steam. We can get to higher temperatures by adding in pressure. So that's where we're talking about the autoclave, okay? Most of the microbicidal effect of moist heat is going to be coagulation, and that happens better in the presence of water. So that's why moist heat is going to be way more effective than dry heat, and I'll show you why. First, dry heat, what we mean is flames and electric coils. Incineration, like we do in lab, right? Bunsen burners. And then we have electric heating coils. We're literally talking about ovens, okay? Actually, literally like the ones you have at home. All right, so this will dehydrate our cells. That'll increase the stability of some proteins, which is crazy to think about, but we have to get to super high concentrations to make it be damaging to the proteins, or high temperatures to be damaging to the proteins. Anyway, so water helps with that so that we can do that faster. And just helps with messing up those hydrogen bonds and everything like that. So without water, the dry heat, without the water, it's not going to happen as quickly, and you're going to need higher temperatures, but not everything can withstand moist heat. So, but yeah, this is mostly going to oxidize things and turn them into ash. You know that, we've seen that. So, right. So, when we're dealing with moist heat, if we took a temperature of 121 degrees Celsius, remember, boiling is 100. So 121 for 15 minutes with moist heat will sterilize. Pretty impressive. For dry heat, you take that same temperature, you don't have moisture anymore, you're going to need to go for 10 hours. That is quite the difference. So even as you get up to 170 degrees Celsius, you're still having to go for an hour. So we know that whenever we're doing our little Bunsen burner thing, that's taking seconds, right? You want to imagine how hot that is? It's way higher than 170 degrees Celsius, right? So we want to kill endospores if we're going to sterilize. That's the goal. And they're going to require temperatures above boiling. So what did I just tell you guys? Boiling does not sterilize. That's what I just told you. So we need temperatures above boiling to destroy the endospores. For the vegetative cells, they can vary. It just depends on the organism. So if you're trying to determine what the best temperature is and for how much time you need to do something. Now, people have already figured this out for us. So we don't actually have to do these things to investigate anymore, but you have to know the concepts, unfortunately. So our terms here we want to know about: thermal death time and thermal death point. So time and point. My notes, when I wrote about this one, I say time to kill for time, and point is talking about the temperature to kill. That's what we're trying to figure out with these. So thermal death time, you have a specified temperature. How long does it take to kill the microbes at that temperature? Okay, so what we're determining is how much time it takes. So both of these are looking at time versus temperature. So the way that point is looking at it, we have a set time, 10 minutes. What temperature do you need to be at to kill everything within 10 minutes? You can imagine 10 minutes is a pretty reasonable amount of time to have to deal with trying to get rid of microbes. You get into 30 minutes, are people really going to stand around and wait 30 minutes for something to work? I don't know, right? So that's what these are asking about, thermal death time. You want to know how much time is needed to kill it at the temperature. Thermal death point, you want to know at what temperature you can kill everything if you have 10 minutes to do it. All right, boiling water is for disinfection, not for sterilization. Do not think you can boil your water and it is sterile. It isn't. The endospores, yeah, the endospores will survive. You'll kill Giardia, but you won't kill the endospores. So this is great though, if you're like just trying to disinfect things for if somebody's been sick in the area, it'll get rid of your viruses for sure. They're not strong enough to withstand that. So if you have somebody that had COVID or something, you can boil things that they had come into contact with to make it useful for the rest of the family again. It would work just fine. But it doesn't get rid of those endospores. Next, we have another moist heat method, pasteurization. This is going to be disinfecting, usually beverages. This is heating and cooling and cycling back and forth. We're going to heat with flash sterilization, pasteurization. We're going to heat for 71.6 degrees Celsius, nowhere near boiling, right? For 15 seconds, just 15 seconds. And then we're going to cool it for a little bit, back to room temperature, sometimes even colder. And they're going to heat it back up again and keep doing that until enough microbes are killed. To say enough, what are we trying to get rid of? Pathogens, really, if we could ideally get rid of as many pathogens as possible. So our batch method will use a much lower temperature, but we'll be cycling at those low temperatures for 30 minutes and then going to cooler temperatures. So when you guys are looking up about storage, I just saw this on one of the YouTube channels that I watched, talking about unpasteurized milk, right? If you guys are talking about unpasteurized milk, you're not going through this to eliminate the pathogens. There could still be pathogens in there. One of the most common endospores that you can see amongst cattle is anthrax. Good luck with that. So, not really. I mean, it just depends on if it like takes hold, right? It can and it can't. It just depends on how it affects you. But potentially, depending if you breathe it in, that's the worst. I think that you can have it. But yeah. I don't think you'd be breathing it in though. But yes, so you want to pasteurize your milk to remove your pathogens. Are we sterilizing the milk? I mean, of course it tells us we're not, but obviously not. Think about the milk that you have at home. If you leave it in your fridge for a month, real milk, not lactate, right? I mean, lactate is milk too, but still. Anyways, you take milk, and it's been only pasteurized and you have it in your fridge for a month, it's going to be chunky and nasty, right? That's not the milk that's doing that. It's the bacteria in the milk that's doing that. So it's fermenting and things like that inside of your milk. Dry, I mean, that should be okay. You can sterilize that. So you can sterilize that radiation. Yeah, they can go for, not want to say forever, but they can go for a very long time. But there's a problem with dry milk too, where you could potentially get endospores back in from the environment if you open it and, you know what I mean, like expose it or whatever. But anyways, so this is great. You have lactate, by the way. Lactate, they add that enzyme lactase to get rid of lactose sugar for people that can't digest it, but they also ultra pasteurize it. So if you guys don't, like I've said, I think I've said this before, but if you drink milk and not a lot of it, I recommend buying lactate because that will last in your fridge easily for a month, easily, without any spoilage. I don't. Wait, I don't know, man. I don't know what to tell you. You can buy the off-brand. You don't have to buy lactate. But yeah, that stuff lasts a really long time. It's not sterile either, but
there's been a lot of recalls lately, even for like alcohol.
Yeah. I probably, well, I would say it has to do with the climate, but it might, I don't know. So steam under pressure. So we did the moist heats that were just, you know, cycling the heat or boiling. Steam under pressure. This is the autoclave. So a lot of us have heard of an autoclave. An autoclave is a device that can subject pure steam, just taking steam and pressurizing it to greater than 1 atmosphere. 1 atmosphere is where we live at. So it's just more pressure than what we live at. And by increasing the pressure, it's easier to get the temperature up. And you don't have to expose it for as long. We get 15 PSI, which is pounds per square inch. That's a measure of pressure. That'll yield 121 degrees Celsius for about 20 minutes. 15 to 20 minutes is enough to sterilize, depending on what you're applying it to. So liquids, like everything we use in the lab, or even your auger, I know it seems like a solid to you, but it was liquid when we poured the plates, right? So we autoclave it at that high temperature. And then when it cools down enough that we can actually hold the bottle, we'll pour out the plates and then they can cool to solid, but they were sterilized by autoclave. This is what an autoclave would look like. I'm not going to go over it, but hey, there it is. There are also tabletop versions of this, which is probably more commonly seen at places like vets and stuff. My lab that I used to work in, we had 4 big ones that we could roll in a cart. Bigger than this opening was bigger than this table. It was a little wider, you know, whole cart that we push into that. Yeah, it was ridiculous. Yeah, it was a lot. And a lot of culture we have to. Yeah. So anyways, we have Bunsen burners. You know that. This is going to be 1870, so 1,870 degrees Celsius. So quite a lot higher than the other. And if you're trying to put things in furnaces or incinerators, that can be anywhere from 800 to 6,500. They're going to use the higher temperature stuff for things like, you know, crematoriums and things like that. So it's faster to burn and more effective.
Do you think they use the highest temperature of the 6,500?
I have no idea. You can ask and let me know. I have no idea. But yeah, so we're going to reduce things to ash and gas. The gas is going to be carbon dioxide that will be released in that situation. What do we say? Not everything, of course, can withstand that high of a temperature. That makes sense. Talk about sterilizing some very delicate, like plastic pieces and things. Okay, so dry heat. Here we're talking about electric coils to start with. So we had the incineration. Now we have the ovens. It is just like an oven. I don't want to make it sound like it's crazy or something, but you can put it in an oven at 150 to 180 degrees Celsius, which I believe is somewhere around operating temperature for our typical, you know, Fahrenheit ovens. But for 2 to 4 hours, depending on what you're dealing with, to sterilize. So you can get rid of things that can't withstand the moisture, like certain kinds of metals. Cold. Let me tell you, okay? If you ever see when in any of these slides where I'm saying some microbes or most microbes, instantly you know it doesn't sterilize, right? It has to be all of them in order to sterilize. We're leaving some things out by saying some. So we know cold doesn't sterilize. We say some microbes are killed by cold temperatures. It's actually not common to be killed by a cold temperature. Y'all put in your chicken breast in your freezer, maybe thinking that the salmonella is not going to be there when you thaw it out. I hate to break it to you, but bacteria can withstand that freezing and thawing cycle.
It's just like doesn't let oil freeze.
Yeah, it just, exactly. So we would say that cold is microbistatic, right? It just keeps them from multiplying. So it does preserve it. It's great for preserving foods, but it's also great for preserving microbes. So whenever we're trying to preserve our microbes for use later, we'll usually put them in the cold. In fact, all of you guys' blacktop tubes and everything, I keep them in the fridge so they stay with the way that they are without other things growing on. Right. So there are organisms called cyprophiles. These guys actually grow at freezing temperatures, so they can become a problem in freezing situations. They actually will still continue to make toxins. I don't know. I never understood that one. That was one I don't actually get. Desiccation, this is the dehydration of vegetative cells directly exposed to normal room air. So that's just talking about drying it out. That's fancy, a lot of words, just saying desiccation. Drying it out. It's not going to affect the endospores. We already know that. They're hardier than that. So some very delicate pathogens, again, the delicate ones can be killed, but a lot of times the pathogens can be preserved. But that's the thing, right? If you're preserving your beef jerky and you dry it out, are your microbes still growing on it? No, because you've dried it out. They don't have the resources they need to grow. So it doesn't really matter too much. That's all you're worrying about. Then there's this process called lyophilization. And lyophilization, I don't know why I'm writing the whole thing, but lyof, whatever, equals cold plus drying. So we'll just take a sample, spin it in a centrifuge in a vacuum to help pull the moisture out of it, but at freezing temperatures. So we're doing that to help preserve it as much as possible. All of our cultures in our lab that you guys have been working with came from lyophilized samples. If you ever seen drugs that you have to add like saline to it or alcohol to it in order to like liquefy the powder that's in there, that's lyophilized typically. So it's useful for preserving things, but it's not going to sterilize. Moving on to radiation. This is fun, right? Radiation is energy emitted from atomic activities. We know atoms are doing things all of the time around them, but whenever they overdo things, they're caused to overdo things. That's when we have the effects of radiation. But radiation itself is typically going to be caused by electromagnetic fields. Okay, so it's like that energy from those waves. If we have waves from photons that allow us to see things, that they operate at those wavelengths, right? But all the stuff on the outside has worth as well. And everything that has more frequency is higher energy. So more and more of those humps. So that includes UV and then X-rays and then gamma rays. And going further out is going to be more effective. So these guys are more excited. I don't know beyond that. There's probably things, I just don't know. So irradiation is referring to bombarding cells or other things with the radiation, right? So subjecting them to those waves that are energetic and causing the atoms within the person, the tissue, the item to become excited. Now, if you're trying to kill microbes, then this could cause damage to their DNA, and so they can't survive that process. So that's true for both kinds of radiation. And there's two kinds, ionizing and non-ionizing. Non-ionizing is UV radiation. So you guys know with UV, so the green ones down here, UV. If you put up a barrier for UV radiation, a wall, let's say, simple as that. Put up a wall between you and the sun. Is it causing you damage right now? No, right? It gets stopped by the barrier. Look at ionizing radiation. It's not going to quit. So it will come through that wall. It will come through almost anything you can put in front of it. X-rays, you can stop with lead. Certain gamma rays, you will not be able to stop with lead. It won't be stopped by anything. So it can cause severe damage. When we talk about gamma rays, we're talking about things that are much higher risk for causing damage than things like an X-ray. Yes, so nuclear reactors, that sort of stuff. And then we're talking about Chernobyl, right? And the whole reason that it's destroyed is the effects of the radiation associated with that.
They found mold actually trying to eat.
Yeah, there's a lot of animals. I've seen studies done showing that animals are surviving pretty well in what's still a pretty highly radioactive environment. I don't know, guys, this sounds like Resident Evil or something. But anyway, so you can see that that's dangerous. If we're talking about ionizing radiation, that's like terrifyingly dangerous. It will actually destroy DNA molecules, break them. It'll also destroy protein molecules. Pretty much anything that you're made of, its waves can damage like completely. So no wonder it causes cancer, right? Because it's just messing up your DNA like altogether. It's not working right. Non-ionizing radiation, it excites the bonds, but it's not going to break up the DNA. We'll talk about how it works in a second, but let's come back to these scary guys, these ionizing. We call this cold sterilization because we can sterilize. Obviously, you'd be able to sterilize with that nasty business. Cold sterilization because we're not introducing heat. You can go pop somebody in front of what they call like the elephant's foot at Chernobyl, which is where the source of the radiation is. Pop somebody in front of that, they're not going to get hot. They're just going to die. So we call it cold sterilization. We actually subject a lot of like things like foods and stuff to this. Most of the time, if you guys get prepackaged like little syringes or whatever it is, we use these a lot in medicine, right?
Yeah.
Those have usually been irradiated. Yeah. Yeah, they're usually irradiated to make them sterile. So we have this in life way more than you probably think. We use it to our advantage, and that's great. I don't know. Let's see. Yeah, it does. I should really think about that. Sometimes I think about that when I'm using something wasteful, and then I just try to push it out of my mind.
Think of the hospital, they're wasting.
They really are. All right, yeah, ionizing radiation, we already mentioned this. We're going to cause ions in the atoms. That's what's really happening there. As we're breaking the DNA bonds, we're causing electrons to be ejected. So we've seen what electrons being ejected, like sodium, when it gave its electron to chlorine, we have Na+, right? That's an ion. Those were ionic bonds. So that's what the rays from the ionizing radiation are going to do. They're going to force ions to be made in places that they should not be made. And then we can't share our electrons the way that we're supposed to in our bonds, and that's why we're breaking our bonds. That's how that works. All right, then the non-ionizing radiation, we're exciting our bonds, our atoms to a higher state. So they will bind next to each other instead of across from one another. So you guys remember how A binds with T and C binds with G. Now imagine if you had a sequence that was like A, T, T, C, G, right? And this was T, A, G, C, whatever. And 5' to 3' and all of that. And these guys are bound. Usually these are bound to one another. So this, what happens is we will excite, usually it's the T's, and they will come and bind to each other instead of to the A's. Because they bind to each other, they can't bind to the A's. These are called, no, they don't.
Oh, they're just floating.
Yeah. And so that becomes a problem. You can see how the structure now of the DNA is, compromised. So those are called thymine dimers. It's not as penetrating. We know that you can put up barriers, but it will pass through air, certain liquids, and not really most solids, maybe like Jello or something, glass if it's not got any treatment on it. But so typically when we treat things with UV, it's going to be the surface. So you'll see people working in like little biological safety hoods or whatever, and then they'll turn on the UV light afterwards. So it's just cleaning the whole surface. Sterilizes, but it takes longer and it doesn't penetrate, not like ionizing. So here's a picture of that thymine dimer situation down here. So how it was to begin with, normal. Yes. Now we have the ability in our bodies to repair this. So if we catch it early enough, if our cells catch it early enough, then they can remove that and like remake that sequence for us. But if we don't, then it stays part of your DNA. And that could potentially lead to the development of cancer, especially if you have a whole lot of that going on in your DNA. That is, yeah, that definitely you're having damage done there. Anytime you go out in the sun, this is happening. Even if you have sunscreen on, you're probably still getting some of this going on. It's just like to a significantly less degree. But that's what those SPF things mean. They're like a measure of how much essentially damage you're taking and exposure to that sun and how long you can be in the sun. Anyways, so then that's going to be a problem whenever you're trying to replicate or go through transcription, right? We don't have the bonds together the way that they're supposed to be. It messes that up.
So is that more like, are we going through revolution all the time, like building, like blood, like molecules?
Yeah, you're always making new cells through like constantly. Yeah. Sometimes more than others, right? Like when you're growing,
Or
but
like experiments.
yes.
You see very small growth when it comes to like hair or nails, but
To you, it's small, but like how many cells did it take to make that what you saw happen? You know what I mean? So look at it that way. And I don't know how long it takes to make a cell. Probably not super long, but I don't imagine it's like 5 seconds or something. Decontamination. This is, we're going to filter things. That's the number one decontamination that we need to know, other than like washing, right? So sudsing things away, sure. But now we're going to filter something. The whole point of filtering is to remove something from another, like the liquid, typically. So if you make your pores small enough in your filter, you can remove any microbe. So you can potentially sterilize. It just depends on size of the pores. We can do this with liquids and we can do this with air, like HEPA filters, right? So we want to do this for things that can't handle heat. So we might do this for milk and beer sometimes. We do this obviously for water purification, and we can remove airborne contaminants. Osmotic pressure is talking about removing, killing, it's more like preserving, but putting things in salt. We learned about hypertonic situations where we have salt all on the outside. So more of a substance on the outside than what would be on an inside. So if we had 95% water in a cell, sound familiar? Now that means you only have 5% solute. If you have 50% water outside, that means you have 50% solute. Where's the most solute? Outside. Yeah. Why are people getting it wrong? That's all you have to do. Where's the solute? Where's the liquid? There's more solute outside. It's hypertonic. Yeah. So, anyways, be sure that when you guys go back to your questions and you see that you missed them, that's one of them. That's a great example. Be sure you know why you missed them, because you will see these topics back again in final. It might not be the exact same question. That's why you should understand them as opposed to just memorize the answer. Because it won't be the same question. I can promise you that. Be reworded. So be sure you understand when you get things wrong. It'll help you learn more than just leaving it and saying, oh, I didn't understand it. I guess I'll never know. Osmotic pressure, salty on the outside, right? If you put like anything, a bacteria into salt, it's going to suck the moisture out. That's what hypertonic does, right? So it's going to cause what we call plasmolysis, which is that shrinking of the cell. It can be deadly, but a lot of times it's not completely deadly. So it's not super effective for like sterilizing, but it is good for preserving. It's excellent for preserving. So most things can't multiply in that, right? It's going to be microstatic. So sugar also works the same way. Cool. Okay. Whew. The chemicals. They can be in all these forms. That's fine. And then aqueous or tinctures. Aqueous, like what it says, like with aqua, that's going to be water-based. Tinctures is a term for if it has any alcohol mixed in to make it effective. All right, so similar sort of situation here. If you're looking at a chemical that's going to be used, these are a lot of the same exact concerns we would have with the physical methods. I'm not really going to go over them too much because I'm not going to go have you guys, list these out. In case you didn't notice, I don't have that on the study sheet. So high-level germicides. These are going to be for your critical items. What does that mean? They're going to touch sterile tissues. So this is catheters, heart-lung equipment, and implants. So we want high-level germicides for that one. Intermediate, these are going to be used for the semi-critical. This is the ones with the mucous membranes, respiratory equipment, and thermometers. Now, catheters, we talk about catheter like a bladder catheter. We might think that's mucous membrane though, but because it's a long tube that someone's pee is just coming out of, and then bacteria can get stuck up in there, it's super difficult to make sure those are clean, and if you have to reuse them, especially, and they do often. So being able to sterilize that, that's why it gets bumped up into the higher level. Respiratory equipment is not as bad. And then low level, I don't know, furniture is mostly what I think about when I think of this, and the equipment that is around and that sort of stuff. Okay. What are you going to worry about when you're using your chemicals? All the same stuff. I mean, how much germ is there? What germ are you looking at? How strong do you need the chemical to be? Is that chemical safe for use on skin or in that application, right? Look at that. That look familiar? This is how these guys are going to affect, same as the others. And guess what? You're going to see that again in the antibiotics as well. We're going to divide them up that way. They just have one more category. All right, let's start with the halogens. This is fluorine, bromine, chlorine, and iodine. The ones that we're really going to talk about are the last two, chlorine and iodine. But do you guys know about fluorine? Yes, of course you do. You guys use toothpaste? You guys drink water? It typically has fluorine in it. And that's to help keep your teeth nice and sparkling and clean to keep from getting tooth decay. So chlorine and iodine, though. So some of the stuff, when I say halogen, but if I say about chlorine and iodine, those are familiar for most people. You know that there's chlorine in pools. And chlorine, you know that probably may or may not, but bleach is chlorine based. So those sorts of things are kind of familiar. We know we use iodine sometimes to prep patients before surgery for their skin. So you can get bromine if you have a hot tub. The only reason I know this is because I have a hot tub. I use chlorine in my hot tub, but you can use it. I see it all the time on the little test strips. That's all I know about it though. Apparently pretty similar to chlorine in its action. But these guys are microbicidal and sporicidal. So this is telling us we kill all microbes, including endospores. That's what sporicidal means. So what is this? What does that mean? Sterilizes, right? We have it in our chart too, if you need to refer to it. So super common ingredient in a lot of cleaning solutions. You have liquid and gaseous forms of chlorine. The chloramines can also be added into water to treat it. They do slowly kill endospores. So if you're just cleaning with something with bleach and just wiping it off, it's not going to probably be effective as a sterilizing agent. But if you let it sit, then it's more likely to work appropriately. But that's what's great about chlorine. It's very versatile. If you dilute it, you can still use it on skin. People even recommend for people who have like certain skin issues, whether it's just inflammation or infections, that you can sit in chlorine baths. And that's recommended. Don't drink it, yes. You can sit in it and expose your skin to it, and it's perfectly safe as long as you're just doing it for like 20 minutes or something and not like living in it like that. But yeah, so it's pretty crazy how useful that can be. We know bleach is a hypochlorite, and it releases hypochlorous acid to denature enzymes, and that's how it would work. I do not have that on the study sheet, turns out. Okay, pay attention to those things. Y'all writing down about how it works and stuff. Not on the study sheet. Look at the study sheet. All right, halogens. I'm not going to ask you about how it works. It's not on the study sheet. Halogens. Oh, right, we're still on chlorine. I thought this was the next one. My bad. So I feel like a lot of these slides will say like the same thing, but with a little bit more added to it. But yeah, we know what we're using the hypochlorites for. We know what bleach is for. It's really useful. And you also see chlorines in a lot of other, like Lysol, but it's not bleach, but it's got chlorine based in there. So iodine, these will, same thing, slowly kill the endospores and destroy all others pretty effectively. We used to use free iodine. We also have iodophores, which are tinctures, meaning they have alcohol with it. So they are pretty effective, but they can be irritating. So we don't use some of them anymore. So these iodophores that we do still use, betadine, povidone iodine, and isodyne, you'll see the first two. I don't know much about the last one, but I see the first two a lot. And they still even offer povidone iodine if you go donate blood and you're allergic to chlorhexidine, which is the other thing that they'll prep your arm with for their draws. They do not prep their arms with alcohol. And you guys know why? Because they use like 14 gauge needles.
Oh, those are good.
Yeah, that's bigger than like my nose ring is 16. 14 is bigger than that. So yeah, they're gonna stab you with that. They want to be as clean as possible. Alcohol is not gonna do it. Anyways, and it's gonna sit in there for like 5 to 10 minutes too. So I don't want to expose you to anything, but they can use iodine still for that if you are allergic to the other thing. So now we're moving on to phenol. This is a different category. We did the halogens, that's chlorine and iodine. Now we're on to phenol, and we have phenolics as well. But phenol itself is also called carbolic acid. It's very effective, but it's not sterilizing. Again, it says some, not sterilizing. That's what we need to know. It will work best in high concentrations, but it's very toxic. It's kind of dangerous to use. So they banned some of them. And now they really only use phenol itself for serious kind of areas like in drains and cesspools and in animal quarters where they really need to get like some, cleaning done. And it can be effective there because of how, strong of a chemical it is on top of it being, effective as a disinfectant. All right. So this was originally used by Joseph Lister to treat his surgical suite. We learned about that, right? So this was the chemical, it was phenol, but it's toxic and irritating. So we have the phenolics, which are versions of it. It just, changed. It's a version of phenol, but slightly altered. And these often have detergent action in addition to the normal, but a lot of these are toxic as well. So I don't know, take it as you will, but none of these are sterilizing. So it is what it is. So the two biggies that you probably should know about in the phenolics, triclosan should sound familiar if you're, over the age of 30 probably, and chlorhexidine.
I've heard of that.
Yeah, that's
That
a very common one.
Sometimes they use that for like, if they have like.
Yeah, it's a really good for cleaning skin, actually, chlorhexidine is. So triclosan used to be added to like pretty much every hand sanitizer and soap and body wash ever that you had, even like dish soaps had it in there. But it's also considered an antibiotic in the way that it works. So it was, causing bacteria to become resistant, and we were finding it popping up in animals that were nowhere near, weren't raised in civilization. They were like in the wild. We were finding high concentrations of triclosan. And so they decided maybe don't do that anymore. Don't have it building up in the environment. So it got banned in 2017. I mean, it was like everything at Bath and Body Works had triclosan in it. It was ridiculous. So it was everywhere. So we don't use it as much anymore, only in healthcare settings. And then chlorhexidine, you guys may know this better at, so yes, I say chlorhexidine, that's what they use to prep your arms and at OBI. I used to work there, so of course I would know that. But Hibiclen, Hibitane, and Peridex. These are soaps, basically detergents that you would be using to surgical scrubs. Use that. Okay? Surgical scrubs have use Hibiclens usually. It's chlorhexidine scrub, and you can buy these over the counter. Chlorhexidine scrubs. You can buy for like body wash. You can use it as body wash or to treat any skin conditions you may have. It's a little bit safer than some other things.
If you get a tattoo.
Yeah, you can use it with your tattoos. I'm personally not advocating it just because I like to just say use, scent-free dial or like something like that for your tattoos. I'm just saying. It's just you can get irritated by this and you don't want that on your tattoo. So and that can cause more problems. But it is very disinfecting if you feel like you may have been exposed to something. You can use that way. So hand scrubbing, surgical prep. And other medical uses for this one. Hibiclens is available over the counter. And like I said, you can take showers with it. If you have skin diseases, you can make a bath out of it if you wanted to. And there's mouthwashes with chlorhexidine as well. Don't need to know what it targets, but hey, it does target the membranes, just so you know. We already said over this. It's a little bit, it's very mild and low toxicity. It's one of the phenolics that isn't as toxic. So that's why we use it so much on antiseptics and stuff like that. All right, alcohols. These will target most, whatever, it doesn't matter, not sterilizing, right? So this is now a disinfectant. All of them are. So a lot of people cleaning with your ethanol or with your isopropyl alcohol, and you think it's sterilizing a thing, and it is not. It is disinfecting, very effective. Concentrations of 50% and higher of these alcohols will disrupt membranes. We already talked about how it disrupts cell walls, right? And the problem with them is that they evaporate very quickly, and they can affect your nervous system with those vapors. They work best with some water. So if you are looking to buy yourself some alcohol for cleaning wounds or around the house, you want to look for 70%, not 100%. 95 and 100% ethanol or isopropyl alcohol are not going to do you any favors. Pick 70, it works better. The water that's present there helps the alcohol to coagulate proteins. So now you're getting cell wall, cell membrane, and coagulating proteins. That's what the goal would be, yeah? Make it as effective as possible, but still safe to use. So 70%, it actually turns out that ethyl alcohol is not as effective as isopropyl alcohol, and isopropyl alcohol is cheaper. So again, I would recommend isopropyl, but ethyl alcohol is a little bit more resistant to evaporation, and so it can be more effective in like the antiseptic and disinfection. Like when we use our hand sanitizers, a lot of them nowadays are ethyl alcohol, ethanol. So it's a little more skin friendly, but isopropyl for cleaning surfaces and wounds and stuff like that, we really need more action, go with the isopropyl. Next, we have the oxidizing agents. So things that are involving oxygen and free radicals associated with oxygen. Organisms that go through aerobic respiration, aero, oxygen, they're used to dealing with oxygen and byproducts that can be toxic, free radicals associated with it. If you're going to breathe in oxygen, you better be able to deal with its, toxic byproducts. So we do, we have things in place that help protect us. And so do certain bacteria that go through aerobic respiration. However, the anaerobics, they are not going through aerobic. They don't see oxygen all the time. And so anaerobics are usually killed pretty effectively by these guys. So if you're treating something in an anaerobic, like a deeper wound or something, this can be really effective. So that's our oxidizing agents. But what are we talking about? We're talking about mostly hydrogen peroxide, okay? That's what I want you guys to worry about. Hydrogen peroxide, surprising yet? How about this? Oh, this part, hydrogen peroxide. What does that mean? Now, the hydrogen peroxide that you use at home is 3%. The hydrogen peroxide that sterilizes is 35%. So you're not getting the sterilizing effect, but it's still pretty effective as an oxidizing agent. Okay, so we'll use that for wounds, mouthwash, and bed sore care as well. Anything that isn't getting a ton of exposure to oxygen, let's just force some of it in there. Again, 35% hydrogen peroxide. We can use this for sterilizing cabinets, or for sterilizing cabinets that we can put things in that don't withstand heat very well and expose it to vaporized hydrogen peroxide. It's actually very good for that. And it's colorless. It's pretty, itself not super toxic, but it's really bad for bacteria that can't handle the oxygen. So let me show you something. This enzyme here, catalase, and this is the last time we're going to see this one. Catalase is an enzyme that converts hydrogen peroxide into water and oxygen. And yes, this is oxygen as in like air, atmospheric, like gas. Okay, so, and then this enzyme catalase is what drives that. So we have catalase, and any aerobe should have catalase because it helps deal with toxic oxygens. They usually get converted into the hydrogen peroxide, and then we deal with that catalase.
That's not bad.
No, it's not. You're right. Well, I'm not going to do that. So forget that. You're right. It is not balanced. So we have for basically, though, this will end up being what your product will be. So the oxygen here. You guys ever use hydrogen peroxide and get little bubbles, little fizzy bubbles? It's your catalase. Your catalase did that. It converted it into oxygen. So that's what's happening. That is a real thing that's going to be going on. And aerobes tend to be able to deal with that. We'll be testing our microbes to see if they can handle it by looking for the bubbles. Let's see, like we said, sterilizing at high concentrations, we'll use vaporized, and that's really, otherwise it's disinfecting. All right, detergents. All right, some. Not sterilizing. So these are our polar molecules that act as surfactants. We already talked about this. So detergents are surfactants. We already know we're going to have hydrophobic and hydrophilic parts to it, right? We don't even need to ask any questions about it because we already talked about it. Turns out that the ones with the positive heads work better. So instead of having where we had phosphate head and the little tail, and this was negatively charged, this is our phospholipid. These guys have a positive head and that, because now they also want to be attracted to that negative. So it helps them work just a little bit better. And the ones that operate this way, these are the quats. So these are quaternary ammonia compounds, very, very commonly used. You probably don't even know that you're using disinfectants that have this in it. Really, really common additive to a lot of like anything you're spraying as far as like kitchen cleaner and things like that. It might also have other things, but quats are usually there as well. And of course, that's going to disrupt your membrane. Do they do well by themselves? No, but they do really well to help other things work better by opening up those membranes. And that's the idea. This is just looking at like the structure of them. We have our positive head. It's even got our little positive here. It's usually N, nitrogen, bound to other things because ammonia is going to have a positive charge to it. Yeah, NH4. So that's what that's basically representing. So we have the same thing down here. This is something maybe sound familiar. Benzalkonium chloride, very skin-friendly antiseptic. I got my ears pierced at Claire's when I was a kid, and that's what they gave me to clean my ears with was benzalkonium chloride. But yeah, so they help with destabilizing that membrane. But they're so terrible at killing all the microbes that they're bad. Someone will grow in them. So Pseudomonas species will grow in soap dishes. If you ever seems like you're getting a film that's not really quite soap or whatever sitting on your soap dish, if you have solid soap, that's really what that is. So this not stopped, man. It doesn't care. Yeah, they're all over the place. To be honest, Pseudomonas is everywhere. Grows everywhere. All right, heavy metals, not like the music. We're talking about some, again, not sterilizing. I don't really care how they come, if they're tinctures or if they're whatever, I don't care. What I want you to know is it's either going to be mercury or silver, really the only ones that we use as antiseptics and that we use for proper, like associated with life organisms. So here's why. You can become resistant to metals, or the microbes can. And it doesn't work against endospores, and you can be allergic to them. But the ones that are more commonly used, silver and mercury. We have mercury used as antiseptics, as well as preservatives in cosmetics, ophthalmic solutions, and other substances. A lot of times you may see that you have mercury in like contact solutions and stuff like that. You just didn't even know it was there, but it's there to keep them clean. So it has its purpose. As long as you aren't like having it in high concentrations, you should be fine. You'll have mercury, by the way, in your environment all the time. It's yes, and yeah, it's all over the place. People just don't get it in a lot of concentration. You should be fine. Anyway, silver nitrate. Probably a lot of us know about the silvers. There's another slide here in a minute that talks about it, but oligodynamic, I'm not going to ask you about that. Yeah, I don't know. They can be toxic, it can be allergic, whatever. Here's our silvers. All right. So, okay, whatever. Take the mercury. Sure. It sounds like it's good for antiseptics and preservatives. Silver, we've got silver nitrate. It's for topical. We've got silver sulfadiazine for burns. I burned myself one time pretty bad. If it gets below like a certain amount, then they'll give you that as a cream to keep like the exposed tissue clean. Pure silver, you can incorporate into the catheters to make them more resistant. Colloidal silver, you can actually, this is a germicide, but you can actually drink this. And people will drink it, and then it can make you turn blue. Look it up, man. People will be like, oh, I'll be, so antimicrobial. Well, you need the microbes in your gut, by the way. But all right, silver ions that we can incorporate into surfaces and textiles. If you buy yourself some scrubs that are antimicrobial, then they probably have silver incorporated into it. All right, aldehydes, we're almost there. Aldehydes, they can be, not all of them are, but can be sterilizing. There's only one of them that is, and that's glutaraldehyde, okay? This just talks about how it works, and I don't care. So glutaraldehyde sterilizes. Yes, and this is, however, pretty like strong. It's very potent. It's not great for like antiseptic use or anything like that. Pretty harsh. Then we have formaldehyde. If it's a water-based solution, this is formalin. It's typically not entirely sterilizing. Depending on what concentration you use it at, it could be, but this is what we use in embalming fluid. And we can use this in some surgical instruments. And then the last one is orthophthaldehyde. Don't try to spell it out. And this works similarly to the glutaraldehyde, but it's not going to destroy endospores, not sterilizing for sure, but it is less irritating and it's faster, but it's more expensive too. So those are options with our aldehydes. The gases, now we have ethylene oxide. There's two of them that you need to know, ethylene oxide and chlorine dioxide. But I think I only asked about one of them. Ethylene oxide is on the study sheet, not the other one. So ethylene oxide, it kills endospores. It's sterilizing. Wow, kills endospores. There we go. The sterilizing gas. Weird, cool. Why are we not just putting everything in this, right? It will act pretty vigorously. It's very reliable as far as sterilizing. It's also explosive and explosive, and it's a carcinogen. And it causes a lot of tissue damage if you contact it. So this is a problem, especially if you're using it the way that they usually use it, which is to sterilize rooms. They can fill whole rooms with this gas, and then they have to be able to pump it out. But people can't go in there while that's still in there. So we usually use it for rooms. That creates something called a chemiclave, which is a chemical version of an autoclave. It's just creating a big room with gas, chemical in it, and you can sterilize everything in there by exposing it. And then chlorine dioxide, which isn't on your study sheet, was used to treat water and also the Senate offices when they were attacked with anthrax. That's 2001. All right, like to put it in perspective, I remember Chernobyl. That was 86. Yeah. Yeah. So I remember, that's how old I am, so it's different. But acids and alkalis, obviously, this one should be a pretty good one for you guys. It's going to involve pH alteration. Again, this is some, doesn't sterilize. If you're interfering with the hydrogen bonds, having too many hydrogen ions or too little hydrogen ions, it can disrupt how the proteins and other molecules are going to be working with one another. So that's how the acids and alkalis work. Some common ones. Alkali, aqueous ammonium oxide. Anything with ammonia is going to be basic. That's an alkali. All right, and a lot of them, you're going to see that in detergents and things like that. And then organic acids. It might sound weird or strange or like, you know, you're learning about chemistry again. So I'll circle a couple of them. Acetic acid and lactic acid probably sound the most familiar to you. We know lactic acid is something that's going to make like your yogurt sour and your kimchi and all of this. But acetic acid is vinegar. We know people use vinegar to clean their houses and stuff like that. It's very safe. It's effective. It's not like the absolute, I mean, it's not sterilizing, but it is safe. It's effective. If kids get around it, they're going to be fine. So for household cleaning, it's pretty good. And that's how it works. And that's it. Whew, let's do the Kahoot. We'll give you guys a minute to breathe, and then we'll do the Kahoots. I'm upset that I didn't get to show you the notes, but if you guys want to see them, I did post them on the modules in chapter 11. So you want to see how I would, like my notes would look like ultimately. And then what I recommend is, as you're taking notes, write down what you need to know as compared to your study sheet, as it makes sense. If it doesn't, circle it and come back to it later and make sense of it. After you've filled it all in to make everything make sense, reorganize it. Reorganize it in like a chart or outline or a brainstorm, whatever you need to make it like make sense to you, to organize it. And then that's all you do. I recommend you do that for every chapter, like after the lecture. Don't wait until before the exam. Just take like 30 minutes to an hour to organize your notes. And then when you come back to take the exam, all you have to do is review those summaries and maybe make some flashcards for some terms that you aren't hanging on to. And that is it. Y'all reading textbooks, y'all rewatching videos and all this stuff. Rewatch things if you really think you need to. If you really didn't get a concept, don't let me stop you, but don't rewatch the whole video just because of one concept. So don't be wasting your time, and I promise you'll do better because you'll be less overwhelmed. You'll be wasting your time less. Stuff that you do look at will be more meaningful and you'll retain it better. I promise. Okay? Thank you. All right, so cool. Just chill. I'll be bringing that up in a second. This is going to be way more fun. I'm going to pause the recording and then restart it. So I can reach the vision. You don't want to see all this. Pause this. Okay.
We're halfway to the end of this month.
I don't know, man. It's happened fast. It'd be a lot happier about it if friggin' computers would work the way that they're supposed to. I've never been so mad at that computer. Whatever.
That was the same room before, right?
Yeah, only for my night class though, usually. I don't usually use it for my Tuesday morning, but they put me in there because they want us to use that room more so that other people wouldn't use it. This is the science building and we were trying to reclaim it back for science instead of letting other people use it for whatever. Yeah, but then we don't have space for when we need to use it. It's a problem. They need to use their own rooms. I don't really care that much. It wasn't my problem. Not the hill that I'm gonna die on. I might die on the hill though of having all the computers set up with Zoom and stuff. So I don't have to keep doing it every time I log on. But yeah.
State was really gorgeous, but when I was in Seattle, it seemed like a lot of the people were unapproachable. Like
Yeah.
I could go into have a heart attack or stroke, and I feel like everybody would just keep walking.
Yeah. Sometimes they're like that. It's really like the South that you see people like friendly and being like, hi, and then don't know you or like, how's it going? You having a good day? And they don't know you. They don't do that. Yeah, maybe. I mean, some people see it that way. I see it as people can be genuinely friendly like that. But yeah, when I lived in the Northeast, it was just like that. I was like you were saying, like people just, they're them and they don't want to have anything to do with anything else outside of.
Well, yeah, because when I was in Seattle, like if you show my friend he was telling me, she was like, if you show any amount of like sympathy or whatever to those who are like homeless, they will not leave you alone. They
Yeah.
will travel you and help you.
Yeah, I don't remember how much I had in Philadelphia. I would have to, I just avoided them altogether. They would like lay on like the steps of the subway. So I'd have to like walk around them. They peed in there and stuff. It smelled like pee all the time. Philadelphia's gross.
Jail was a little bit. I literally saw a guy smoking crack. Just broad daylight.
That sounds about right, yeah.
Then I went to another area and there was like, those line bikes. I saw one of those like just in the lake. I didn't.
Yeah, when I was going to grad school, when I go to like some certain, depending on where I was in Philadelphia, but like I would go to Chick-fil-A, it wasn't Chick-fil-A, it was KFC. It was
Okay.
KFC. And KFC, and they had bulletproof glass, and they like open up this thing and like shove like the food through that, and you can only have like one side, like a freaking bank teller or
Are
something.
you serious?
Yeah. Yeah.
Wow.
That was in I think that was in West Philadelphia. That's where I lived, but
Was it dangerous?
yeah, it was dangerous. I mean, I heard gunshots. My apartment got broken into, but I didn't really mind it. If you knew,
You don't mind your apartment getting broken?
I hated that part, but yeah, I didn't really mind the rest of it. As long as you knew like what was safe and where to go and everything, it was fine. And on campus, it was great. All right, let's do this. Someone just kicked in my door, my apartment.
Wow, they must have been very strong.
And they stole my scooter. Well, they didn't. They didn't drive it off because they couldn't figure out how to drive it. They just left it in the street, but. Alrighty,
Glad you got it back.
yeah. So, safety, lab safety. I have on the, I reframe my mind here. On the notes that I gave you guys, the study sheet, right? If it's not on there, I'm not going to ask about it. It's just like any other study sheet, okay? So there's a few safety rules, even though our safety rules sheet is like 17 rules or something. There's like 8 or something on that. I'm going to ask about maybe 1 or 2 tops. So, but those ones, none of those are hard. I wish that all the questions were as easy as, you know, this one is going to be. All right, where should broken glass be disposed? Right. So, I mean, those ones will be easy. I wouldn't worry about it. Don't bother even trying to study for it, I would say, because you know what you need to do. All right. For the stains, I know each stain has its little section, and then I made a little summary of the stains. So you'll have to know everything that's in each section, but at least you have that summary to like compare to see how they're different and whatever. So I would go over that as well. But you need to know the steps in order, right? Each stain that is used and its purpose. What is each step doing for the stain? Okay, because it all has a reason. What is positive and what is negative as far as how it will look like? Because I'm going to show you a microscope, and it's going to have a slide on it, and it's going to be a question of like what's the primary stain used in this procedure? And I'm not going to tell you what the stain is. You're just going to have to know that, oh, this is the endospore stain, so that primary stain is malachite green or whatever. So that's how the questions are going to be on the test. I feel like it's pretty, to me, the questions are straightforward as far as like what I tell you guys to know, but don't get lost in just knowing the literal things, be able to understand it. Like the last part, this is what I'm saying. What happens if you skip a step? Even if I don't go over every single step with you on all of these, just be able to visualize or write it out on your scratch paper. What would happen if you skipped a step on this procedure? Okay.
Saffron will always be a counter stain, right?
I believe so, or a simple stain. All right, what is the purpose of heat fixing? Okay, so we're going to kill it and adhere it to the slide. That's the goal. Yeah, I don't know, just those 2 things. It itself is not going to be what, stains it or anything like that. Just killing it and adhering it to the slide is what it's important for. What is the counter stain used in the gram stain? Okay, so the counter stain in the gram stain is going to be safranin. I would say just know in general that stains where they're going to work is primary. They may or may not have a mordant. They may or may not have a decolorizer. And then they may or may not have a counter stain. And they'll always be in that order. There's one that has a secondary stain and not one of the other things. All right, what color will gram negative bacteria be after the gram stain? All right, so this is red. Red's a negative color to me. That's how I remember that one. So that's going to be the gram stain, either red or purple for that one. What I was going to say, on the exam too, I have 2 questions about, I think that are relating to gram something. So like one of them may be like, this is, gram positive, blah, 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 blah question. But the other one is going to, oh no, I don't think it does say, but it doesn't matter. There's 2, usually I have one that's positive and one that's negative, and they're usually right next to each other. So if you look into it and you're not sure if it's purple or red, because the red is pink and sometimes it has a purple shift to it, like it looks kind of purpley. So if you're not sure, wait till you see the other one and I bet it'll answer your question. I bet, because usually that purple is purple and it'll not be, anything you'd be questioning. And you can always ask questions. What is the first stain used in a capsule stain? Alrighty, so capsule stain, this is the one with the little halos. So those, we needed to have a background, if you will. And Congo red serves as the stain that allows us to do that. I'm pretty sure the next question is going to ask us about what I'm getting at here, but so Congo red for that one. What type of stain is Congo red? Pick all that apply. I gave one of them away. Okay, so it is a background stain, like we said, but it's also a negative stain. That negative charge of it is repelling the negative on the surface of the cells. And so that's what makes it stain the background instead of the cells. So they'll repel each other. All right, this is a pure culture. What's going to be pictured is, what stain is this? This is pure. This is only one kind of bacteria. Alrighty, so this is the endospore stain. I think it's going to ask about the red cells, red structures. All right, what is the term for the red structures in the image? Right, so those are the vegetative cells. You will, even in endospore, because that was a positive, right? That's the example of what a positive looks like. So the positive, even whenever they are making endospores, will also still have vegetative portions of their cells. So you'd be looking for red cells with the little green structures all around. Usually pretty obvious. You're not going to just be a couple. Then the negatives should be all red, and that's it. So you're just looking for the presence of the little green guys amongst the red. Right, which of the following bacteria would be acid-fast positive? Okay, so this is Mycobacterium. Anything Mycobacterium will be acid-fast positive. And in fact, the only thing that's going to be acid-fast positive is Mycobacterium. So it goes both ways. Mycobacterium, all of them acid-fast. All the acid-fasts are Mycobacterium. And that's it. So none of the other ones will fall into that. That means that they have a waxy coating, right? So what is the primary stain that we used in the acid-fast stain? Okay, so this is carbofuscin for this one. So it's our primary stain. The first one, the one that's deciding basically positive or negative, carbofuscin. It is lipid soluble, so it integrates with that waxy coating. That waxy coating, again, what's it made out of? Mycolic acid, right? That's why it's Mycobacterium. All right, in which staining procedure did we use steam? Alrighty, so this is going to be the endospore stain. We actually would consider steam to technically be a mordant because it's helping that stain to actually stick inside of the endospores. So it will apply that term, but I'm not going to refer to it as that on the test, but it's just to help you guys understand how it's working. So which of the following structures allow for bacterial motility? Select any that fall into the category. All right, great job, guys. I don't even have to question it. It's only the flagella. All right, so we didn't talk about simple stains in those questions, right? But simple stains, we know heat fix, easy enough. And then we're going to put one stain on it and then look at it from that, right? In our example, I used methylene blue. There are a bunch of others you could use. But if I were to tell you that you did a simple stain with methylene blue on, yeast, if you did simple stain and methylene blue on gram negative, you did simple stain, methylene blue, gram positive, all of those guys are just blue, everybody blue. That's all it does. Okay. It's not affected by anything else, by any of the other stuff, but it can help you see structures that would allow you to tell the difference between things, potentially. Anyways, gram stain, that's going to be based on the cell wall thickness, mostly. Acid fast, that was the mycolic acid, waxy, and that's why the carbofuscin is lipid soluble while we use that one. The counter stain for the acid fast is methylene blue, though. The capsule, that was our primary stain was Congo red, right? And it allows negative stain, get the background. But then we added Manivals, right? And we didn't talk much about it, but that is a secondary stain. And that's why we don't have a red background. That's why it's kind of purpley. It's from the Manivals. It changes the color of it. So that's why, and it lets us see the cells as well. And that's why we use the Copland jar, because we did not heat fix that one. Heat fixing gives false halos, right? So wet mount, that's just looking at motility. We're not going to see the flagella, but that is what they use to move around. And the other thing about the wet mount, Brownian, no, correct, no heat fixing, but Brownian motion. Know what that is. It's just water molecules pushing the cells around. They are always moving around doing stuff, atoms. But they're going to bump around the cells, and that's why they look like they're shimmying around the whole time. So you want true movement, not Brownian motion. And then we have endospore stain, which we just talked about, malachite green. We used the paper on it, yeah? The paper was just to keep everything wet, like the wet blanket theory. So it doesn't do anything else. It's not a mordant, it doesn't do anything else. It's just to keep it wet. And then that's malachite green, 10 minutes over the steam. That's what's really doing the work there for us. And then we will rinse it with water. That was like our decolorizer. Yeah, even our vegetative cells get rinsed clear, hopefully. And then we counter stain with safranin again. So ELISA, know this test. We say inside and out. You're going to need to know the theory of this. I'm not going to show you an ELISA result. You need to know how it works. So what does the E stand for? All right, so this is enzyme linked immunosorbent assay. What is an indirect ELISA looking for? What's the question it asks? Ready. So most of us got this, at least we were leaning in this direction of either antibody or antigen. So that's going to be the one that we're going to have to, I guess, focus a little bit more on. But the antibody, that's basically something that we make in response to having been sick with something, right? It's not the thing that made us sick. it's what our body did to react to it. So that's what we're looking for in the indirect, because if we were looking directly, we would just be looking for the thing. And that's what the direct ELISA does. It looks for the antigen. So at least we're on the antibody antigen. That's what drives ELISA, how it operates is antibody antigen specificity. All right, that was one question. What the principles of how the assay works. This is talking about you would start with antigen or antibody, depending on the thing. Then the patient sample, then secondary. So those two at least are always there. And then adding the substrate to see if it cleaves it to make a color. So it's colorimetric. That's the gist of it. Antibodies are proteins made by the body in response to specific antigens, right? And antigens are anything that your body, your immune system can respond to, see as foreign. All right, indirect, like we said, you're going to be looking for the antibody. Has this person ever seen this thing? And direct, we're looking for the thing directly. How the color change happens, the enzyme, if it's there or not, if it's positive, it will be. If it's negative, it won't have made it through that whole process of rinsing and everything. We didn't get into rapid testing in our class. That was my last class. All right, I mean last semester. DNA stuff, yay. All right, what does PCR do? Its basic function. Okay, so PCR amplifies DNA. It just makes copies of DNA. That's all it does, is just make the copies. And remember, you guys are going to get asked that question. If you start with 1, how many will you have after 4 cycles, or 5 cycles, or 6, whatever it is? Never more than 6, huh? So it doubles every time. So if it was 1, if we started off with 1, after 1 cycle, it would be 2. If we went another cycle, it would be 4. Another cycle, 8 from the previous one. Yeah. It's not like adding 2, it's multiplying. Does that make sense?
So if we did 3 cycles and times that by 2.
You times the number 2's result by 2.
Oh.
So it's like multiplying by 2 every step from the
Okay.
one previously. Yeah. So it's just like, I mean, it really is just doubling it. Let me see. There's no markers in this one. It's doubling it. It's not meant to be. Let's see if I can draw it. You have 1, and this is double stranded. Okay, but I'm just not going to draw it that way.
Yeah.
1 becomes 2. I should say 2. Hold on, I'm thinking about this. 2, right? The 2. This was after 1 cycle. This was after 2 cycles. And then 4, 6, 8. That's 8. I mean, 3.
That's like
3. That's the 3rd cycle. Okay. And then if there's 4, you would that, because each one makes 2. If you need to draw it out on your scratch paper. That helps. But yeah, each single thing that we started off with is going to become like 2. So each one becomes 2. So that was 1. That one became 2. And that was 1. It became 2. So that was 4.
if you went through 2 cycles, then you have 4.
Yes. So you could do the math from here if you wanted. So if after 1 cycle, you had 2, you doubled that, right? You could say, okay, well then the second cycle would be double this, which is 4. The 3rd cycle will be double this, which is 8.
So 4, yeah, 4 times 2.
Yeah, multiply by 2, the previous one.
I was gonna trick that.
Yeah, don't overthink it. Alrighty, which DNA fragment is largest? Please read all your choices. Okay, the largest one is going to be 35 kbp, which is 35,000 base pairs. So we've got 35,000, we've got 100, we've got 4,000, and we've got 1,200. And if you guys need to write it out when you see it, you can do that. Nobody's going to look at your scrap paper. All right, which of the following DNA fragments would travel the farthest on an agarose gel? So it's gonna be 100. Good job, guys. That's the smallest one. Smallest travel the fastest. All right, what type of cells are capable of taking up exogenous DNA?
I love that picture.
All right, the term for this is competent, or you could say that that is essentially the definition of competent. Competent cells are able to take up exogenous or outside DNA. All right, so DNA structure and function. I'm not really going to test you on that. The basics of it will help you understand the rest of it though. All right, so DNA fragment sizes, that's just talking about the kilobases and base pairs and all that stuff. So don't let the Ks. Dude, I am standing. What are you talking about? My phone, my watch says that I need to stand up. Boy. All right, agarose gel electrophoresis principle. DNA has a negative charge to it. So it's going to migrate to the positive end. And then we have the smallest ones traveling the fastest as well, because they don't get as stuck up in there as trapped. PCR word, how it works. And it says in a practical sense. I just need you to know what each of those major 3 steps are, what's going on in each of them. So we have denaturation, heat, we're splitting the DNA apart. We have annealing, which is sticking things together. So anneal the primers onto the parent strands or whatever. And then we have elongation, just adding the nucleotide. And that's it. That's all the steps in PCR. And you just cycle through those over and over again. And again, be able to do, tell me what number you have after whatever number of cycles. It's not gonna be more than 6. Okay, bacterial transformation. Know the steps, I'd say the steps, but the steps were easy as far as like the transformation part, right? All we did is put the plasmid with the bacteria and put it in the hot water and it took it in and then we sealed it up with a cold. That was easy. The media though, you will need to know what will grow and what will be produced on each media type after that, okay? So what happens on nutrient agar, what happens on nutrient agar with ampicillin, and what happens on nutrient agar with ampicillin and arabinose. So, and what's being made at each one. And that chart that's on that handout is correct. I don't know, it'll have that information on there. And you won't have, I will ask about that, and you won't have the plates. So just so you know, like some people like to work with that, but I'm not gonna give that to you for the exam. Plasmids, what are they? Remember, plasmids are DNA. Yeah, circle. Typically, they can be eukaryotic, but typically they're going to be bacterial. Circles of DNA, not necessary, carry extra genes. And that's what we use to manipulate our cells. All right, eukaryotes. I say review your drawings. You guys probably took pictures or you're looking at my pictures that I gave you. That's fine. Which organism is this? This is probably Giardia. Nope, it isn't. That's definitely not Giardia. Which one is this one? You guys remember? Pink paintbrushes. So this is either Penicillium or Aspergillus. I'll warn you guys that Enterobius was one of the helminths. So we should know at least that that belongs in that category. So we really only looked at Penicillium and Aspergillus as fungi. And those structures that we looked at are called conidiophores, right? It says that on the paper. But yeah, FYI, you'll need to know that. So penicillium and aspergillus. Penicillium, the way I remember is P for penicillium, P for pink or purple, whatever you want to call that. I don't care. Pink. P, penicillium is pink, paintbrush shape. Okay. The other one, aspergillus, it's like greenish teal. That's what it looked like on you guys's, and it looks like dandelions. Like the puffy ones. Okay, so like I said, remember the differences. And in case you can't tell on that question, then you're going to be asked a similar question on the lab exam. Know the difference between aspergillus and penicillium. You don't need to know if those are going to be your choices. There'll be 4, just like there was there, but those are going to be the only real options. Similar to the protozoa, right? I told you guys about that one. What is the name of this organ? Probably Giardia. So this is Giardia, FYI. But yeah, you guys, yours will not be Giardia, and it will not be Entamoeba histolytica, and it's not going to be, I don't know, Enterobius or any of that. It's going to be either Trichomonas or Trypanosoma. Those are the only ones that you're going to see for the protozoa. Okay, so know those differences. If you guys are like, look at me like, oh man, how much to know those differences? Look at the pictures between Trypanosoma and Trichomonas. It might be small on you print it out, but if you zoom in on it, they look just drastically different from one another. We're talking about red blood cells with little purple, snaky looking guys. That's Trypanosoma. Or we're talking about I don't know, they look sort of like Giardia, like pink, but they're going to be bigger than Giardia. They'll be like 40x. So, FYI. Right. Organisms that you had to draw or take pictures of, that you have pictures of. Focus on the differences, okay? Important structural and reproductive differences. This is really what I'm getting at here is we learned about fungi, we learned about protozoa, and we learned about helminths, right? Which ones of those make eggs? Helminths, only helminths. Maybe you know that, okay? Concept of trophozoite versus cyst. A trophozoite is like vegetative. Trophozoites are the active dividing forms. So I think troph to me sounds like trough, like you're eating. Okay. So you're not eating when you're dormant. So that just like with the endospores, the cysts are our dormant party structures. All right. Like I said, differences where you want to focus on and transmission. Do be aware of how the helminths are transmitted because it does, like that's one of the tables that was on that worksheet. Yeah. If you probably, just know how they're transmitted. Okay. Yeah, so general stuff about bacteria growing on plates and stuff and isolating them. Oh, which is not a term used to describe colony morphology. So only one of these doesn't belong. Dude, you're saying that I have not been standing, but for 1 hour today, you got to get out of here. All right. Hyphae is the answer for this one. So we could go through each of these and I could tell you what they all mean, but I'm not going to test you on what they mean. What you guys really need to know for this one, and the reason why I didn't even write this out on the study sheet, is know what terms are colony morphology terms. Know what terms are cell shape. Which ones are cell arrangements. So hyphae is the branching structures that fungi make. The reason they have those branching structures, hyphae. Anyways, I will probably give you a list and you pick the one that is the thing. So be able to pick it out. That's what I'm saying. It won't be that hard as this one. All right, what is the name of this type of culture? So notice that this says auger at the top of it. Okay, so this is a slant. A lot of people pick broth for this one. I don't want to call anybody out, but does anybody want to volunteer why they picked broth for this one? So that I can at least understand, because people pick it a lot. I called broth, but what did I call those black top tubes that I gave you guys? They're, because when you stand upright, they still stay like that. Yeah, because of why? Because they're solid. And broth is broth. Yes. Yes. So a lot of people think that those pictures is just liquid on its side and I just put it upright. I don't know where people are thinking that from, but yeah, that's literally what slants look like standing straight up, right? The black top tubes that you guys have are slants. So be aware of that. They're slants. Solid can only be auger or slant or butt. It can be butt. That's the thing. Broth is always liquid, always liquid.
I love that.
Yeah. Yeah, we'll deal with butts later. We haven't seen them yet, but they're coming. All right, cool. Because a lot of people were missing that one. All right, how was this organism isolated?
Faster than like foreplay or serial solution.
No. I mean, they exist, but we're not going to do them. It's just too much work. So yeah, so the two that we need to know for isolation are streak plate, the quad streak. That was the quad streak, right? And the serial dilution, which we didn't do, and I don't have any pictures of it, but it is on like your study sheet thingy. Serial dilution is like if I take like one of the broths that we had, like one of those liquids that are like super, just a ton of cells in it, and then dilute it out 1 to 10. And then if I were to plate that and there's still a ton of colonies and I can't get individual ones, dilute it 1 to 10 again and plate it. And that's serial dilution, just 1 to 10, 1 to 10 until you get one that you get individual colonies. It's more for counting, but it is used for isolation as well. Just so you guys know. All right, which arrangements of bacteria do you observe? It really, I'm gonna warn you guys, it's just gonna be one. Okay, so this is strepto, strips, chains. That's exactly what we were seeing. It did also have diplo. That was one of the things that was on there. It wasn't true bunching as far as like, I understand that it folds back on itself, but that's not actually true staphylo arrangement. It's just folded back on itself. So
Staphylo's more.
yeah, it's going to be pretty obviously bunches. What is the name of the shape of these bacteria? I guess maybe I did take something. Oh, no, I didn't. That's right. He's supposed to be over there. Never mind. Yeah. This is, yeah. Okay, so these technically are spirillum. That's fine if you got this one wrong, because guess what? In the lab, we're only going to have bacilli and cocci. If you know the difference between those, you're gold. So I won't give you anything besides that. It's just on the list of some of the shapes of the cells, but we never have them in labs, so I don't focus on it a lot. But yeah, it is a thing. Umbonate is a colony morphology term. Okay, so colony morphology, recognize the terms as being colony morphology related. Media types, we have the solid ones that are auger and slant, and then we have the liquids that are broths, and then we have the butts that are semi-solid. They're semi-solid, but isolation techniques, we really only have to worry about focusing on the quad streak. I won't ask you about the serial dilution, but quad streak, know it. Okay. Cell shapes, cocci or bacilli, those are your only answers. We don't have any other kinds of bacteria in there. And then cell arrangements, you're not going to see anything except for staphylo or strepto. I don't have anything that does anything else. So, okay, microscopes. If you observe a specimen at 40x objective and have a 10x ocular lens, what is the total magnification of the specimen? All right, good job, guys. This is in fact 400x, it's just multiplying, yeah. I am gonna have a microscope that is set up. It might be even one that you have to look at a slide in. But you will have to tell me what its total magnification is set up. And I would expect you guys to be able to pick these numbers off of the lenses appropriately.
So
Do the math.
will they do like total, or will there be like just for the objective one? What is that?
So what objective lens has the number on it? Why would I ask that? This is a kindergarten.
That's right.
So no, yeah, it'll be set at, let's say it's at 40x. Then what would the total magnification be? You guys remember what it would be? Our oculars are always 10x. So it's 400. Okay. It says it on our oculars as well, in case you don't remember. You can look at it, it's fine. You can look at it, turn it if you need to see it, it's okay. I don't have a few rules for the lab exam, but that's not one of them. So which objective lens do we use for oil immersion in our lab? Okay, so this is our 100x. I don't want to say it's going to help with preventing light scatter because the oil is more similar to the glass. Glass to air, that air in between causes the refraction, the light to scatter. So we add oil to keep it where it's supposed to be. So magnification, be able to calculate it. It is okay to look at the eyepieces. Sometimes the eyepieces, because they can come out and stuff, you might have to rotate them a little bit. Just be gentle when you do it. If you needed to. I don't know why you would, but I'll try to make sure that it's where you can see it. But anyways, and then the objective lens, you'll have to like look at that for that question. But yeah. Understand how the microscope operates, what's happening with the light and the lenses, all the lenses, where's the real image being made and the virtual image being made. Remember, real is where we're first seeing it. That's going to be our objective. And then virtual is what we get out of the eyepiece, right? The ocular lens. Know all of the parts and how to use them. Most of them are pretty straightforward. Things like if I were to ask you about the condenser and the diaphragm might be things that not everybody is familiar with, but most of the other parts you guys probably would be. So just focus on the ones that you might not be as familiar with when you're studying. Understand why we use oil immersion. I think I already explained that. So prevent light scattering. Okay, Kirby Bauer. Which antibiotic is this bacterium least susceptible to?
Good picture.
Yeah, I did not take it.
I did that wrong. I did not see E.
That's okay. Yeah. E, I don't blame you for that because E was it was overgrown. It was not looking for circles. Yeah. So E, that's the one that's completely grown everywhere. It's the least susceptible. So if you're reading that in the question, let's say, and get to that, and it's asking which is the least susceptible, you can break it down into whatever simpler terms that you like not sensitive to it. If you can write that down to remind you, okay, what's the one that's not as sensitive? And then find that so that doesn't words sometimes, right? So if you need to, most of you guys did fine with it, but yeah, remember, you also need to be able to look at it from the resistant side, like which one's the most resistant, which would have been E. It was completely resistant. All right, remember, ZOI is zone of inhibition. It literally means that zone where growth was inhibited, so the clearing area. So that clearing area where there's no growth, at the edge of it, we would tell us the minimum inhibitory concentration, that's MIC. That's essentially the amount concentration of that antibiotic, the K antibiotic, let's say here. It diffuses and gets less and less and less the farther away that it gets. The concentration at the edge is the minimum inhibitory concentration. It's useful for determining how to treat your patients. We're not going to calculate it. That's just the definition of the term. Mueller-Hinton auger, we use it because it has even thickness and the pH, is balanced so that we can let the antibiotics do the job the way they're supposed to, diffuse the way they're supposed to. and be compared from plate to plate because it's not different thicknesses and things like that. And then we have bactericidal versus bacteriostatic, one of the more easy to understand terms, I think, as far as at least the terminology. So cidal kills, static inhibits, right? If I were to give you one of these plates that had the discs on it and we have ZOIs, and I asked you, can you tell if this antibiotic K killed these bacteria? The answer would be,
We don't know.
we don't know yet. Exactly. You know that it's inhibiting the growth. That's all we know. We would have to sample in that area and put it on a new plate like we did in lab in order to determine if that K antibiotic is bactericidal or bacteriostatic, whether it kills or just was inhibiting. That's based on no growth or growth, respectively. Yeah. Okay. I think that's it. So I'll show you guys your little podium so we can all, celebrate our lives. Happy studying. Let me know if anything is confusing when you guys get to studying. It's 50 questions, multiple choice only. It's going to be on Scantron, so please bring pencils, number 2 pencils that have reliable erasers. I have some, but if you want a reliable eraser, I can't always guarantee that. So feel free to bring your own there. No notes or anything, just like usual. It's just going to be Scantron and that, and you will get a scratch paper.
So I'll have lecture and
You got for the exam, yes, you will need the lab coats. Good question. I consider that part of knowing the lab safety, essentially. So yeah. No, I forgot it again. Forgot it in my other class too. I guess I gotta forget it in all of my classes now. So let's just give everybody credit for this day.
session.
Yeah.
Sorry, I am tired. Literally the day after, like the next day, I had to go to work from my.
Yeah.
And then I have a quick question. It wasn't brought up on the slideshow. I don't know if we need to know it. With the electrophoresis plates, does it go from negative to positive or positive?
So how the DNA goes?
Yeah. Like how does it travel from positive to negative or negative?
It's going to be negative to the positive.
Okay.
Yes.
That's what I was trying to remember. I could.
Yeah, that's fine. Yeah, it'll be moving towards it. So yeah, the negative would have to be at the other end of it. Yep.
You started yours? Do you know what you're talking about? I do
I'm not gonna mess this with last week. I don't think that I can still do it.
I have
Answer the
stuff.
questions.
Um,
It's due by tomorrow, like due date.
yeah, I mean, I have stuff to allow you to do it. I just don't have time to do it
I
today.
know, yeah.
So, yeah,
I can do it later in the week. If that's okay with you. I don't really care. It doesn't matter to me, but if you don't, it's fine too.
yeah, I don't mind if you want to, everybody's results were the same on it. It was like the positive control, obviously purple, the
Right.
negative clear, and then it was like purple clear, light purple clear or something like that. And then you could answer the questions just based on that, but yeah. It does ask about like diseases and things like that. And that
Okay.
part I didn't have. I got it all together and I didn't bring it, so.
It's okay. Don't worry
Yeah.
about it. Don't worry about it. I just wanted to see. And then I was looking on, I just wasn't thinking last night. So as far as, we are literally covering the entire chapter on that immunity, right?
Yeah, that's how a lot of people's are like. I was gonna talk about that with you, actually,
I
for
pulled
part of
up
the thing.
the study sheet, which answered a lot. And has Jennifer Lopez responded back to you?
No,
She just talked to us and she said she hasn't started yet. She
I don't have a clue what's going on, probably.
Oh, was she here
She said,
today?
did you get that email? Yeah, she literally said, oh, yeah. Did you get that email? I said, yeah, the one Brittany responded to.
Yeah, the one
said,
that
"Oh, you
the
already-"
second one.
She said,
that's
oh,
that
you
one?
already did it? I said, no, I mean, I did it. I didn't turn it in. Brittany turned it in. This is Brittany.
Yeah. Okay, by the way, this is your partner.
Well, it basically already goes over what I was thinking about looking at the study guide.
Yeah,
this
yeah.
morning. So,
Yeah, so what I said, and we can talk about that if you want, I'm trying to bring it up. So I took the study guide and I basically broke it down like, not this is the, here's the study guide. Because Brittany did this part down here, vaccine and
Right.
natural immunity and all of this. So we have 3 other sections. The top one, antigens, and I would say break it up as either just going over antigens or over the APCs and MHCs, because that's a lot. That'll just.
Yeah,
that's
what
what
we
we
were
were
wondering.
thinking about. Mine kind
Okay.
of rolls over
Into
to the
what I was-
immunity.
And you
so
don't have to cover everything.
Yeah, that's what we
So-
were basically wondering. Okay,
Yeah.
so on yours, Brittany, is the immunity?
I'm active. I'm the active,
Literally these last.
Yeah.
Ones, yeah.
Okay, so
Um,
that's that. And so this will be 1. This will be 2. And then you're saying these 2 together or?
well, so here's what I say. Topic 1, probably antigens would be the better thing to go over.
Okay,
Just because in order to understand the rest of it, and you might have to talk about MHC and the
bit,
other ones,
mhm.
but just mention them.
Okay.
You don't have to go into too much detail. Cover the basics of these.
okay.
So just like the basic outlines, don't worry about like exact little details of them. So,
So cause you did okay, topic 4, so I can,
Kind of rolls into.
yeah,
Or could
well,
I
will
will
do.
if I do like B cells and antibody,
yeah,
2
Mhm.
cells. So then you'll do,
yeah.
and you're gonna do B cells and antigen? Rachel, is that what you said?
B cells and antibody.
Antibodies. Okay, so basically we need to figure out what she's gonna do.
Well,
I
you
would-
figure out what you're gonna do.
yeah.
And then you can say, this is what's available.
What's
And
the
if you're
harder
not
one?
gonna
'Cause I'll do
accept
the harder
The
one.
harder one is the T cells, I think. If you
Oh, I
put-
love the T cells. Thank you
Oh.
for saying that. 'Cause I already love them myself.
Yes, so we'll put you onto T cells and we'll just have her
Okay.
worry about antigen. So
Okay.
hopefully she can handle that. That's not
Okay.
too much of a monster.
Okay, well, how do you feel about B cells and antibodies? Is that a monster?
I don't think it should
No,
be.
it's not.
Yeah,
B cells are so easy. I still have notes from even anatomy and like, they're so easy. They're so
Okay.
easy. They're so easy.
I was thinking like the
B cells are the
immunosuppressants
memory
and stuff.
cells that remember everything. It's so fun.
Oh, do you want B
I'm
cells?
such a
or
dork.
Becky?
No, I'm just a dork.
Okay.
I just love this. I love this section.
So yeah, I
Alright.
think that'll be fine. And
So-
remember, just go over the basics.
Will
Like,
you,
Well,
What
I
you
was
need to
freaking
know.
out.
will you send that to us?
Yeah,
Just so
I'll
it's,
just send you a picture.
so it's a little better broken downish.
And this is just the study guide that is on here.
Yes, I was just saying
Just
like.
a little
how it's
FYI.
labeled.
I just looked at it this morning. I was freaking out. I'll send it when I get home.
No, you're totally fine. Mitch.
Okay, all right, that's it. That's it. That's all we really needed. Okay.
Perfect. That was
Cool.
quick, fast and.
yeah.
on to the
And
next.
I was gonna tell you on your app,
Mhm,
one thing I forgot to tell you, because I noticed how you're writing.
mhm.
You can change your pen. Oh, you
Somebody
did change
look
it to
your stuff.
Yeah, I kind of go
Because
in between.
They will, it will keep reading. Sometimes it will delete what you write because you're like, I have a problem, I guess, picking up my pencil
Mhm.
and it would keep deleting.
It
your
does,
shit.
it does mine occasionally, yeah.
I was so annoyed.
When I draw the arrows like I've been doing, every time I'd make an
Oh.
arrow, it will delete it.
Okay,
So,
on that, you can take your pen out. If you want to do an arrow and you want it to be straight line, go to this one. And then it also has an option where require hold and then fill in the color. Okay, so like I'll undo it, but you can circle it. And as long as it's all together, it highlights it that color. And you can undo if you don't want the circle and you just want it highlighted. I know that's stupid, but whatever, I just thought I'd tell you.
Yeah.
Because it's nice. 'Cause
yeah,
I'm weird,
there's
I
a
like
lot of
that.
it that I'm still just figuring out. so
No,
it's
you
good.
will. They're like, I just figured, I still learning stuff and I'm like, oh.
Yeah, and I just got this pencil,
noticed
the
you
the
used
pencil is the Apple Pencil Pro, and it got more features. I didn't realize that a pencil could have more features
No, it
without
does.
buttons on it.
And
I can squeeze it and stuff, and it does respond to
see,
that.
I have the old iPad, but I don't have the option, but hey, use this. I'll give you one of these. These are cheap as hell. Use this instead of that, and it will be fine fit, and these fit with everything, so all you do is unscrew it and screw it back.
Oh, wow.
I know it's stupid,
I
but
was
it
thinking
works.
about buying. stuff
Well,
like
they're
this.
cheap. They're so cheap, and
Yeah, I saw they were
literally,
like $5 for like 4 of them.
like this one I've had for an entire year, but I thought these would be different, but
Yeah.
they're not. So, anyway, it'll make it where you can actually, I don't know, it's not as fat, I guess you would. say.
Yeah, yeah.
Okay.
Alrighty.
Thank you for your help.
Is that all you wanted to talk about?
Yeah. I mean,
Okay.
I was just really freaking the fuck out last night because I haven't done anything and
It's
I
a
know,
lot.
I called Rachel and I was like, I just want you guys to know I'm not trying to be an asshole, like, I'm literally just really overwhelmed right now.
Yeah, yeah, I think that it's less than, it's less, like I was trying to say in the emails, like, I think it's less than what you think this is gonna be, but
It's really overwhelmed.
yeah, I think finally, though, that Brittany at least is like excited about doing the work. I don't know why this is what it took. I don't really care. At least she's doing it.
That's
Yeah.
exactly what I said. I'm over it. I don't care. I
Yeah.
just want to get it done.
Yeah. Yeah.
Whatever.
Cause what she gave, it's fine. I mean, it's not like as much work as I would put into
No,
it,
I
but,
know.
but it'll be fine,
It,
so.
it worked.
Mhm.
It worked. And I'll edit it, whatever.
Yeah. So at least
Sorry, Joe.
at least she's helping, I guess.
No, I'm not. Right.
Yeah,
What do you want to talk about? That's it. I'll get everything ready.
right,
Just tell me what you want to talk about. So,
yeah,
no, it's not a big deal. I'm just whatevering.
it's okay.
And, mom, they, I don't even know what they're doing. But I read the report. They still haven't, she's not even gonna go see an oncologist herself. Wednesday, I think it is,
Mm-hmm,
but it says that if it says it's the malignancy, that is where